All right, so good evening once again. So the last time we finished up with coenzymes, and that was the very last lecture that we had on the fundamentals of enzymology. Today we are starting with metabolism of biomolecules. I told you earlier that we'll be doing metabolism of carbohydrates, metabolism of proteins, lipids, and nucleic acids. So we will start today with the metabolism of carbohydrates. Now, right from even primary school, we have always known, we have been taught that carbohydrates serve as the source of energy to the body. Today, we are going to build on such knowledge, even from the senior secondary school, from the second cycle education we had about the metabolism of carbohydrates, where we have known that energy is derived from the simplest form of carbohydrate being glucose. So in other words, glucose, serves as a source of energy to living organisms. At the cellular level, of course, that's going to be energy to the cells. And then at the level of the tissues and organisms, you have glucose serving as that source of energy to the whole organism. Now, we also will see that, or we have known that plants, or let's say photosynthetic organisms, have the capability of preparing carbohydrates. So in the process of photosynthesis, you have plants, being able to synthesize carbohydrates. But for non-photosynthetic organisms, such as human beings, we cannot synthesize these carbohydrates. So we get these carbohydrates from plants. So plants become the source of the carbohydrates that we get to give us energy in food. So we have in plants these carbohydrates stored as starch, and in some cases, some other sugars like maybe sucrose. So you have these carbohydrates stored in either the roots of the plant, which we go for as root tubers, or it could be stored in the stem. For instance, sugarcane will have carbohydrates stored in the stem, and we have in some plants carbohydrates stored in the leaves, some stored in the seeds or fruits. So you can get some amount of, say, sugar, fructose in some fruits. And in some cereals like, maize, millet, sorghum, rice, you have carbohydrates stored in these plants as starch. But in animals, we have these carbohydrates stored as glycogen, and glycogen could be synthesized in, or could be stored in the liver, or we have glycogen stored in the skeletal muscles, so that in times of fasting, so for instance, at night when we are sleeping, you have the body, especially the brain cells, falling on energy that is stored as glycogen in the liver. If the body is involved in some vigorous activity and needs some quick energy in addition to the normal, metabolic processes already occurring in the cells. The body, the muscle cells fall on the glycogen that is stored in the skeletal muscles. So we are going to look at 5 different pathways of carbohydrate metabolism. First is going to be glycolysis, which you have studied even at the HND level, and the TCA cycle, also known as the citric acid cycle or the Krebs cycle. These two, I'm sure you have seen, but we will be paying attention to some details that perhaps at the HND level, you have not paid so much attention to. Then we'll look at glycogenesis. That is the pathway for the synthesis of glycogen, that storage path. Then we'll look at glycogenolysis. That is the breakdown of glycogen. So you have the carbohydrates stored as glycogen in animal cells, either in the liver or in the skeletal muscles. And when this storage form is fallen upon to give energy, the process by which the glycogen is broken down to give energy to the body is what is referred to as glycogenolysis. And then we'll look at the last pathway, which is the pentose phosphate pathway, which goes into the synthesis of nucleic acids and even some nicotinamide nucleotides, which will go into some synthesis of lipids and like some oxidative, biosynthesis. So these are the pathways that we'll be looking at as far as carbohydrate metabolism is concerned. Now, if we take carbohydrates and specifically that simplest form of carbohydrate, glucose, from which the body gets energy, glucose can undergo these 3 different pathways of utilization. Of course, these are not the only pathways. As we go forward, you see that there are further pathways which are extensions of these, extensions of the glycolytic pathway. And then we will also see the extension of the pentose phosphate pathway and the storage pathway. So we will see that glucose can either undergo these 2 catabolic pathways, that is the breakdown of glucose. So that the first here is glycolysis, the breakdown of glucose to give us pyruvate. And it doesn't end here. Pyruvate goes further into the citric acid cycle or the TCA cycle or the Krebs cycle. And glucose can also undergo again this oxidation pathway. It's a breakdown to give us a 5-carbon sugar, which is a ribose 5-phosphate. And the name quickly should tell you this ribose 5-phosphate goes into the synthesis of nucleotides. So specifically, ribose 5-phosphate will go into the synthesis of nucleotides, which will go into the synthesis of RNA. If it is going to go into the synthesis of DNA, then of course there will be some process of reduction. That will be the loss of oxygen on the 2nd carbon of the ribose to give us a deoxyribose. And that will then go into the synthesis of nucleotides, the deoxynucleotides, which will go into the synthesis of DNA. And then if there is some excess some excess carbohydrates present in the living organism. If it is in animals, it goes into the synthesis of glycogen, and that will be glycogenesis. And if it is in plants, it gets stored as starch, as I mentioned earlier, starch in the roots, starch in the stem, starch in seeds, in fruits, or even in the leaves. And you can have some carbohydrate also stored in the form of sucrose, or sucrose, of course, that will be in sugarcane, and you have some which will be fructose, which you find in some fruits. So what we're going to do going forward is we are going to take these pathways one after the other. So first we'll take the glycolytic pathway, and then after that we'll take the pathway of the synthesis of glycogen, which is similar to that synthesis of starch, and of course sucrose is a disaccharide coming from glucose and fructose. And so the application of that knowledge of the synthesis of glycogen will give us an idea of how starch will also be synthesized, or sucrose, which is just coming from two monosaccharides, will be synthesized. So we will focus on the process of the synthesis of glycogen. As I mentioned earlier, we're looking at glycogenesis. So specifically glycogenesis as far as the storage pathway is concerned. And then we'll look, the very last pathway is going to be the synthesis of ribose 5-phosphate, which is the pentose phosphate pathway, which will be the very last one that we'll look at to end today's lesson. So let's go forward, and then we'll look first at glycolysis before we look at the storage pathway, and then we'll continue from there. So this is glycolysis, which you had studied earlier. Here you see the first phase of glycolysis, made up of 5 different reactions. And this first phase of glycolysis is referred to as preparatory phase. And in some cases, this phase is also referred to as an investment phase. And that will be understood as we progress. You will come to understand why it's sometimes referred to as an investment phase. Now, you see from the beginning, glucose, that is the simplest form of carbohydrate, which is to be broken down. Remember, glycolysis is a catabolic pathway, the breakdown. Of course, coming from the name glycolysis. So lysis, lysis means to break down, and glyco is coming from the word glucose. In some cases, that glyco, just refers to, sugar. I mean, I think that will be from a Latin or a Greek word, which is glycos, meaning carbohydrates. So breakdown of carbohydrates, or breakdown of sugar. Let's say breakdown of sugar. That is what glycolysis means. So we see glucose being that primary molecule which is to be broken down. And the first thing which happens here is a phosphorylation reaction. You remember I told you that in many metabolic pathways, you find that the very first steps are phosphorylation reactions. And such phosphorylations are a form of activation of a molecule to go down a reaction path. And so you are seeing that here again, where you see the very first reaction which is occurring, referred to as a first priming reaction, is a phosphorylation reaction where a phosphate group is transferred from ATP onto glucose to give you glucose 6-phosphate. Now, 1 here refers to the enzyme which is catalyzing this reaction. And you see the enzyme which is catalyzing the reaction is hexokinase. So from our knowledge of enzymology, again, we saw that kinases are enzymes responsible for the transfer of phosphate groups. And from the name hexokinase, you can also tell that this enzyme has group specificity. Group specificity in the sense that it will transfer phosphate group onto all hexoses. That is all 6 carbon sugars. So you can deduce that from what we did earlier on the fundamentals of enzymology. So you have glucose 6-phosphate. So, I mean, when you look at the right-hand side, you also see the structures of the various molecules where you see glucose. So glucose molecule is here, and glucose 6-phosphate is here. So the phosphate group on the 6th carbon is what you see here. So you now have a glucose 6-phosphate. Now, this glucose 6-phosphate now is isomerized to fructose 6-phosphate. You remember when we were doing the fundamentals of enzymology, I think either in our first or second lecture, we saw these 2 structures, glucose 6-phosphate, fructose 6-phosphate. And together in class, we counted the number of atoms of the various elements which were present in the structure of these compounds, the glucose 6-phosphate, fructose 6-phosphate. These, the glucose 6-phosphate, fructose 6-phosphate. And we counted the number of carbons, we counted the number of hydrogens, we counted the number of oxygen, and then, of course, the phosphorus. And we came to the conclusion that glucose 6-phosphate and fructose 6-phosphate were isomers. They are isomers. And so you see the enzyme which catalyzes this reaction, the second reaction, is phosphohexose isomerase. And so that's an isomerase responsible for isomerization of the glucose 6-phosphate to fructose 6-phosphate. Now, we move on to the third reaction, where you see another priming reaction where you have a transfer of ATP, a transfer of phosphate group from ATP onto fructose 6-phosphate to give you a biphosphate. You already have 1 phosphate group, so if you have a second phosphate group added, you now have a biphosphate. So you have fructose 1,6-biphosphate. So you see, this was the first phosphate group. Yeah, that's the first phosphate group. And you are now having the second phosphate group on the first carbon. So you have first carbon and the 6th carbon carrying the phosphate group. And so you have fructose 1,6-biphosphate. And the enzyme that is responsible for catalyzing that reaction, again, is a kinase. And so you see the name is phosphofructokinase 1. Now, the 4th reaction, this is where actually the lysis or the breakdown of the molecule happens. This is where you see the lytic reaction. And so you see it being written there as cleavage of 6 carbon sugar into, 3 carbon sugars. So you see the shaded portion here. This shaded portion is what you see here. And the unshaded portion is what becomes this compound. So you see the bond here is broken. The bond here is also broken in that 4th reaction. And so you have 1 carbon, 2 carbons, 3 carbons in that shaded compound. So you have 1 carbon, 2 carbon, 3 carbons. And the second portion, which is unshaded, also gives you a compound of 1 carbon, 2 carbons, 3 carbons. So glucose, which is a 6 carbon sugar, is now broken down into 2, 3 carbon compounds. And you see the first compound there is glyceraldehyde 3-phosphate. And of course, again, from first year biochemistry, you know what the aldehyde group. And even currently, you are doing functional group chemistry. So you can tell what an aldehyde group is. That's an aldehyde, and thus it is what? Glyceraldehyde 3-phosphate. And the second compound is dihydroxyacetone phosphate. So what you see, the dihydroxyacetone phosphate, the enzyme which is responsible for that reaction is being referred to as an aldolase, which is coming from the word aldehyde. Because of the formation of aldehyde, that enzyme is being referred to as an aldolase. Now, in the fifth reaction, you see that the dihydroxyacetone, the unshaded compound, is converted to glyceraldehyde 3-phosphate. So in the end of the first phase, what you have is 2 molecules of glyceraldehyde 3-phosphate. You have the first molecule of glyceraldehyde 3-phosphate, and you have the second, which is coming from isomerization of dihydroxyacetone phosphate into the glyceraldehyde 3-phosphate, so that you now have 2 molecules of glyceraldehyde 3-phosphate. You see, all what happened to the dihydroxyacetone phosphate is that you have this ketone group converted to a hydroxyl group, and then you have this CH2OH, group also converted to an aldehyde group. So you have some, oxidation occurring on this carbon, where you have the CH2OH group, call it an alcohol group, converted into an aldehyde group, and you have a ketone group that gets reduced to a hydroxyl group. That is what has happened. So you can say you are having both some oxidation and reduction reaction occurring at the same time, but actually this is an isomerization reaction. If you look at the dihydroxyacetone phosphate and the glyceraldehyde 3-phosphate, they are isomers. And so you have that enzyme that is responsible for that reaction being also an isomerase, that is a triose phosphate isomerase. Triose because the compounds, the substrates are 3 carbon compounds. And of course, when you read biochemistry, you see that these trioses, specifically glyceraldehyde 3-phosphate, is referred to as the smallest carbohydrate, which is a triose. A triose meaning is made up of 3 carbons. But of course, in itself, the glyceraldehyde 3-phosphate in itself is not a carbohydrate, but in the process of the metabolism of carbohydrates, specifically glycolysis, you find that the glyceraldehyde 3-phosphate, as well as the dihydroxyacetone phosphate, are some intermediates in that process of the breakdown of glucose, and thus it's sometimes being referred to as the smallest form of carbohydrates, trioses. Right, so you keep in mind that the very last product here in the very first phase is glyceraldehyde 3-phosphate. And you also keep in mind that in this first phase, there is an expenditure of 2 molecules of ATP. The first molecule was in the first reaction. This is where we have the first expenditure of ATP, and we also have the second expenditure of ATP in the third reaction. So 2 molecules of ATP have been used in the first reaction. Keep in mind the process of metabolism of carbohydrate is to generate energy for the body. And here you see energy is not generated, but rather energy is being used. And so that is why you come to see why this preparatory phase, or phase 1 of glycolysis, is being referred to as an investment phase. A process which is supposed to generate energy is rather making use of energy in the very first phase. So this is an investment of 2 molecules of ATP. And we'll soon see what these investments of these 2 molecules of ATP will yield when we move forward to look at the continuation of the process. So we continue from glyceraldehyde 3-phosphate from the second phase. So phase 2 of glycolysis is being referred to as a payoff phase. So if there was an investment, then there should be a payoff. There should be a stage where you have some dividends coming from the investments that you have made. So we have the glyceraldehyde 3-phosphate. Remember, now we have 2 molecules. From glucose broken down, we now have 2 molecules of glyceraldehyde 3-phosphate. So the very first reaction you see here, even though it is a phosphorylation reaction and at the same time oxidation reaction, I want to point out the fact that the phosphate, which is being transferred here onto glyceraldehyde 3-phosphate to give us 1,3-biphosphoglycerate, is not being transferred from a molecule onto the substrate. It's not transferred from a molecule. It's an inorganic phosphate which gets conjugated onto the substrate to give us the product 1,3-biphosphoglycerate. And thus you see that the enzyme which is catalyzing this reaction is not a kinase. You see the enzyme which is catalyzing the reaction is glyceraldehyde 3-phosphate dehydrogenase, not a kinase, because it's not transferring the phosphate group from one molecule to another. It's a dehydrogenase because that enzyme is catalyzing an oxidation reaction. As we saw earlier, oxidoreductases are also referred to as dehydrogenases. So oxidoreductases are those enzymes that catalyze oxidative reduction reactions. Right from the beginning, when we were doing the fundamentals of enzymology, you can go back and revise that. So you see both oxidation and phosphorylation occurring in this 6th reaction. And you see the nicotinamide, dinucleotide, which we studied as a coenzyme, responsible for also oxidative reduction reaction, doing some transfer of electrons and acceptance of some hydrides. You see that happening over here, where you have NAD+, accepting some hydrides to have that oxidation reaction occurring in this 6th reaction. And so you see there was a phosphate group on glyceraldehyde 3-phosphate, and you see a second phosphate group which will be on the 1st carbon, and so you now have 1,3-biphosphate. 1 on the carboxylic. This is almost like from, say, the aldehyde group which was here, the hydrogen is now being replaced with a phosphate group, and this phosphate group on the 3rd carbon was already there originally, and so you now have a biphosphate. Then we see that the 7th reaction now gives us 2 molecules of ATP. So that is a payoff. Remember, we spent 2 molecules of ATP in the first phase. Now 2 molecules of ATP are produced in the 7th reaction. So we can say this molecule of ATP, which is produced here, has paid off the two molecules which were, spent in the first phase. And as you see, this time around, there's a transfer of phosphate group from the biphosphoglycerate onto ADP to give us ATP. And that's the enzyme in that seventh reaction is a kinase. So you see that that is a phosphoglycerate kinase. Now, we also see right from the seventh reaction to the eighth reaction, this is, some form of isomerization, 3-phosphoglycerate to 2-phosphoglycerate. What you see here is the phosphate group on this third carbon moves to, there's that interchange between the phosphate group and the hydroxyl group, so that you now have the phosphate group coming onto the second carbon, whilst the hydroxyl group goes onto the third carbon. So this is what you see here: hydroxyl group on the third carbon, phosphate group on the second carbon. But this time around, the enzyme is not named isomerase, but it's a phosphoglycerate mutase. I mean, this is just coming from the word mutation. It's like there's a change. So sometimes when you see enzymes also, named as mutase, the reactions that you see such an enzyme, catalyzing isomerization reactions. So it's just that the isomerization, the enzyme that is catalyzing the isomerization reaction is being named after the word, mutation. That's why you see the word mutase over there. But it's actually some isomerization reaction which is occurring. Then the ninth reaction, the last but one reaction in glycolysis, you see there is the formation of an enol. So an enol is say, you have alkene, that is these hydrocarbons with C double bond C bonds in them. So the alcohols of such alkenes, alkenols, are then being are called in a short form as enols. So as you see the double bond, the double bond is being formed between the third carbon and the second carbon here. And though you see that there is also some dehydration occurring, and so of course that hydroxyl group is lost. But because of the formation of this double bond, you have the enzyme which is catalyzing this ninth reaction being referred to as an enolase. Now we go to the very last, from phosphoenol pyruvate. The phosphoenol pyruvate, you now have again the very last formation of two molecules of ATP. So there is that transfer of the phosphate group on phosphoenol pyruvate onto the adenosine diphosphate to give you adenosine triphosphate, ATP. And in all these cases, remember we are dealing with two molecules, and so you see 222 in brackets aside these molecules. And so you are having phosphate group coming from two molecules, and that's giving you two molecules of ATP. Remember, two molecules of ATP used in the first phase has already been paid for in the seventh phase, and that's it. This last molecule of ATP is now a dividend. So there was an investment of two molecules of ATP, and we now have, 4 molecules of ATP. And so you hear that the gross product, the gross amount of ATP which is produced in in glycolysis is 4 molecules of ATP, but the net, that is, if the 2 molecules of ATP which were used in the first phase are deducted from the gross, the net is going to be 2 molecules of ATP. And you see the very last product in that process of glycolysis is pyruvate, and that is coming from here. The loss of the phosphate group here now results into the formation of a ketone group on this second carbon, and you now have the 2 molecules of pyruvate being the final product in the process, in that process of glycolysis. Now, before we move on, let me draw your attention to the fact that the nicotinamide nucleotide, which is also produced here, its conversion, this can be converted into ATP. I mean, when you look at if you are looking at, say, the electron transport chain where you have the synthesis of ATP, the amount, the 1 molecule of the nicotinamide nucleotide is equivalent to 3 molecules of ATP. And if that is the case, it means if you have 2 molecules of NADH, this is equivalent to 6 molecules, because this is going to be 2 times 3. 1 molecule of NADH is equivalent to 3 molecules of ATP, and that's, if you have 2 molecules, then you are having 2 times 3 molecules of ATP, which is 6 molecules of ATP. And so you also see that in biochemistry books, sometimes when you read, it's also being referred to that in total, if you are looking at, you are considering the amount of the nicotinamide nucleotide, NADH, which is also produced in the process of glycolysis, then it is deduced that the gross amount of ATP which is produced is, so we have 2 molecules produced here, another 2 molecules, which is 4. And so if you add that to the 6 molecules, it means you are having 10 molecules of ATP if you are considering this amount, the amount of NADH, which is also formed. But then if you take into consideration the 2 molecules of ATP which have been spent in the first phase, then of course that's going to be left with 10 minus 2, and so you have a net of 8 molecules of ATP, which is the product of the energy, the total energy which you are getting from the process of glycolysis. Right. So then we move on to the fate of pyruvate. So remember, the very last product here is what? Pyruvate. So what happens next to pyruvate? What will happen to pyruvate? Because the pyruvate, the reaction, the energy reaction, which is, the whole pathway does not end with just pyruvate, but the pyruvate continues in the metabolic pathway. So yes, what will happen to pyruvate? We see that in the process of glycolysis, yes, we already saw that glycolysis comprises of 10 successive reactions, and the very final product which we saw was 2 molecules of pyruvate. Now, these molecules of pyruvate can undergo different pathways. Now, in anaerobic conditions, you can have the process of fermentation, or you can have the process of what is referred to as lactic acidosis. Now, what is that process of fermentation, where you have the production of ethanol? So let's go back so that we'll be looking at a structure so we understand it well. So if this is the structure of pyruvate, fermentation comprises of decarboxylation, where you have this carboxyl group, the C double bond O, removed as carbon dioxide. And then you have a reduction of these ketone groups. So you will then be having CH3, CH2, OH, and that will be ethanol. So this carboxyl group removed, that is decarboxylation, and then you have a reduction of this group, which will then give you an OH group. So you have a CH3, CH2OH as alcohol. And that is what you see here, that you have ethanol and then carbon dioxide. So in what is referred to as anaerobic conditions or hypoxic conditions, you have the production of ethanol. And this is a process which is, used even in industry for the production of ethanol from carbohydrates. So that process of production of alcohol from carbohydrates actually, is coming from here, where you have glucose. Yes, glucose is broken down all the way to pyruvate, but when it is in an anaerobic condition, you have that pyruvate then going into the production of ethanol. And then you have lactic acidosis, which occurs in the muscle cells, in erythrocytes. When you are involved in a very vigorous activity, what it means is that you are the rate of burning of carbohydrates to generate energy is fast. And so if it so happens that the amount of oxygen which is inhaled in is not as much as needed for the amount of carbohydrate which is being burnt to give energy to the muscles, you have what is referred to as muscle pull. That is scientifically lactic acidosis, or the synthesis of lactic acid, which is also known as lactate. So in that anaerobic condition, so going back to the structure of pyruvate, what happens is that you now have a reduction of this ketone group, so that you have this ketone group converted to an OH group that becomes lactic acid. Nothing happens to this carboxylic acid group. That's why it's an acid. And so you have this carbon on this carbon, this ketone group now becomes an OH group. And so you have a CHOH on this carbon to give you lactic acid, which is also known as lactate. That process is what is referred to as lactic acidosis. But the major pathway which we'll be looking at is in aerobic conditions, how energy is generated in animals and in plants, and even in some microbial organisms. So you have, first of all, decarboxylation, and so you see carbon dioxide coming out. Again, if you look at the structure, yes, decarboxylation, you have this carboxylic acid group being removed. That will be the decarboxylation, as a byproduct, carbon dioxide coming out. That will mean we'll be left with the acetyl group, that is CH3 and then C double bond. So this becomes some acetic acid, but that will be an acetyl group, to which you have coenzyme A conjugated to give you acetyl-CoA, and this acetyl-CoA goes into the citric acid cycle, what you also know as the TCA cycle or the Krebs cycle. And in that process, there is further breakdown, there is further generation of energy to give the byproduct carbon dioxide and some water. And then energy is generated. So we'll continue to look at this process of the citric acid cycle, where you have the production of more energy and the byproducts, carbon dioxide and water. So this is the Krebs cycle, which you have seen severally, many times, right from first year till now, first year of HND till now. And you see the acetyl-CoA, which we are talking about here. This is acetyl-CoA going into the citric acid cycle. So here you see the acetyl group, to which you have the coenzyme A conjugated to give you acetyl-CoA. And you see in the very first reaction, you see here as acetyl-CoA enters the cycle, this is oxaloacetate, a 4-carbon, intermediate. So 4 carbon, this is 1 carbon, 1, this is 2, this is 3, and this is 4. 4 carbons. So if you have acetyl-CoA, which is having 2 carbons coming to get conjugated to oxaloacetate, citrate will then be having 6 carbons. So you see 1, 2, 3, 4, 5, 6 carbons. So that is a conjugation of 2 molecules. One is having 4 carbons, the other is having 2 carbons to give you a total of 6 carbons in citrate. So clearly, this is not a breakdown. This is a build-up. And so that first reaction is an anabolic reaction. Is the production of a bigger molecule from smaller molecules. And so that process is anabolic. And the enzyme which is responsible for catalyzing that reaction is citrate synthase, coming from the word synthesis. I mentioned earlier that synthesis or biosynthesis refers to anabolic reactions, refers to the build-up of bigger molecules from smaller molecules. Now we see that from the 6-carbon molecule, you have some removal of water molecule, which is a dehydration process to give us what is referred to as cis-aconitate. Now the enzyme which is responsible there is referred to as aconitase. And you see that as water is lost, now this hydroxyl group is removed, a double bond is formed on that carbon. And you also see that some hydrogen, 1 hydrogen is lost from this carbon, and so you are left with only 1 hydrogen over there. Now, subsequently, you see that there is some reverse of this dehydration. You now have a hydration, and so this process is a reversible reaction. You see that there's removal of water, and here there is that addition of water. Just as you see here, there is some water in that reaction. There's some hydration occurring, and here there is dehydration. And so this is also reversible reaction occurring, just as this is also a reversible reaction. But still, the number of carbons are the same: 1, 2, 3, 4, 5, 6. No carbon is lost. But if you look at the fact that there is some dehydration occurring, which means some, atoms of elements is being lost from the molecule, that is a breakdown. And so it means you are having some catabolic reaction because water moves out, the molecule is not the same, the molecular mass is smaller. Some elements, some atoms are lost from that compound, and so you are having a breakdown. If you have, again, hydration, you are having a build-up again. So you see why I'm referring to, build up and then there is breakdown, for you to see that this process of, the TCA cycle or the citric acid cycle is having both anabolic and catabolic reactions at the same time. And because of that, this process is referred to as an amphibolic process. Amphibolic process. Coming from, amphibians. These are animals living in water and on land at the same time. So this, because of the two processes occurring at the same time, both anabolic and catabolic reactions. And so you have the TCA cycle being referred to as an amphibolic process. Now we see that there is some decarboxylation occurring. The first decarboxylation reaction occurring here, so it's being referred to as an oxidative decarboxylation. Then, of course, there should be a reduction in the number of carbons. So you now see 1, 2, 3, 4, 5 carbons. There were 6 carbons. Now we have 5 carbons because of the decarboxylation reaction which occurred here. Some amount of carbon dioxide is lost. So it's given out. That is a byproduct which you see, in the process of the breakdown of carbohydrates. And there is that second decarboxylation also occurring here. From alpha ketoglutarate, you have here another decarboxylation. So from 5 carbons, you are now having 1, 2, 3, 4 carbons. So in succinyl-CoA, you now have 4 carbons. Now, we also see that there is some production of ATP here. So you see ATP. In some organisms, it will be GTP, guanosine diphosphate. So in place of adenine, there will be guanine. So you have ATP, and you also, will have, this dissociation of coenzyme A from succinyl-CoA to now give us succinate. And so on that carbon where you have the coenzyme A, attached, once the coenzyme A is dissociated, you are left with a second carboxylic acid group. That gives you that 4 carbon molecule succinate, which is 1 carbon, 2 carbons, 3 carbons, 4 carbons in succinate. You also see that the next reaction, you have some, dehydrogenation, and then you have the production of what is referred to as fumarate. All along, you can be looking at the names of the enzymes, and you see that the names of the substrates are being used. So when it's a dehydrogenation reaction, you see it is succinate dehydrogenase. When it is a synthesis of coenzyme A, you see, because it's coming from succinyl-CoA, this is succinyl-CoA synthetase. And when it is some dehydrogenation of alpha ketoglutarate, some call it decarboxylation and oxidation reduction reaction at the same time, you have that being referred to as alpha ketoglutarate dehydrogenase complex. Now, we also see that when you're having hydration of fumarate, the enzyme being responsible for that, named after the substrate, is fumarase. And then you have malate. So all these intermediates now have only 4 carbons in them because you have 2 decarboxylation reactions. One in this third reaction, decarboxylation, carbon dioxide is out. And then in this fourth reaction, another carbon dioxide is out. And so you are left with 4 carbons in these intermediates throughout up to oxaloacetate, which we refer to in the beginning of the reaction where you have it combining with acetyl-CoA to give us citrate, which is a 6 carbon molecule. Now, look at this. You see that this cycle, even though is based on just 1 molecule of acetyl-CoA, remember, 1 molecule of glucose gives you 2 molecules of acetyl-CoA. So you remember that 1 molecule of glucose gives you 2 molecules of pyruvate at the end of the second phase of glycolysis. And that pyruvate is what goes to after there is decarboxylation of that pyruvate, and then there is that conjugation with coenzyme A, you have acetyl-CoA, which means 1 molecule of glucose gives you 2 molecules of acetyl-CoA. So this whole cycle, this is just for half of glucose molecule. So anything which you see here, if you want to know the amount of that you get from glucose, you have to multiply by 2 together, because this is just showing 1 molecule of acetyl-CoA. But if it is 1 molecule of glucose, everything here is times 2. Now, generally, we know that metabolism of carbohydrates should generate energy. That's what we started talking about right from the beginning. But so far, we have talked about the synthesis of ATP here. This is the only place we see that there is some ATP produced. And of course, because I just mentioned that for 1 molecule of glucose, you have to multiply by 2, which means so far we have seen just 2 molecules of ATP being produced in this FIT reaction. We have not seen ATP produced anywhere again. But you remember that I mentioned that NADH is equivalent to 3 molecules of ATP. 3 molecules of ATP. So if this is equivalent to 3 molecules of ATP, this is some form of energy which is being produced here. And you have in 3 different reactions, this molecule of NADH being produced. The 3rd reaction, in the 4th reaction, and in the 8th reaction, you have NADH being produced. So which means that is 3 molecules of NADH. And remember, for 1 molecule of glucose times 2, which means 3 times 2, you have 6 molecules of NADH from 1 molecule of glucose. And if you are having 6 molecules of NADH from 1 molecule of glucose, this will be equivalent to what? 6 times 3 molecules of ATP. So you really will have to sit down and do that calculation to come to an understanding that in the citric acid cycle, this NADH is equivalent to 18 molecules of ATP. You have these 3, right? So I'm taking it again. These 3 reactions giving you NADH, meaning you are having 3 molecules of NADH. But remember that this is for just half of the molecule of glucose. And so if it is for the full molecule of glucose, it's going to be times 2, which means these are 3 molecules times 2 will give you 6 molecules of NADH. And I said 1 molecule of NADH is equivalent to 3 molecules of ATP, which means this molecule of NADH, which is being produced here, is equivalent to 6 times 3 molecules of ATP, which is equal to 18 molecules of ATP. That is coming from NADH alone. All right, so 18 molecules of ATP coming from here, NADH. Remember, if you add it to that 2 molecules of ATP here, you already have 20 molecules of ATP. How about FADH2? Now, 1 molecule of FADH2 is equivalent to 2 molecules of ATP. And again, this 1 molecule of FADH2, this is for just half of the molecule of glucose. And so if it is for the full molecule of glucose, then it means this is times 2. So you have 2 molecules of FADH2 coming from 1 molecule of glucose. And if you have 2 molecules of FADH2 coming from 1 molecule of glucose, this will give you times 2. 1 molecule is equivalent to 2 molecules of ATP, which means this is equivalent to 4 molecules of ATP. Plus the 20 already, then we're having 24 molecules of ATP being produced in the TCA cycle. That is, if you do the calculation of the conversion of NADH and FADH2 into ATP, in total, you are having 24 molecules of ATP produced from the TCA cycle. Now, we are done with that breakdown of the carbohydrates in that glycolytic pathway, going all the way to the TCA cycle. Now we take the path of storage, the synthesis of glycogen. Now, the beginning of the synthesis of glycogen, which is being referred to as initiation, is in two reactions, where the first reaction, in fact, these two reactions, the result of it is that you have the conversion of glucose 1-phosphate. This is glucose 1-phosphate being converted to glucose 6-phosphate. Now, remember glucose 6-phosphate we saw right from the beginning of the glycolytic pathway. So what it means is that that glucose 6-phosphate, right at the beginning of glycolysis, if it so happens that there is enough energy already for the cell, that glucose 6-phosphate will then go into the process of the synthesis of glycogen. It will go into storage. It will go into the process of glycogenesis. So it can either go down the path of glycolysis or it goes to storage, which is into glycogenesis. Now here we see glucose 1-phosphate that you first have the enzyme phosphoglucomutase carrying a phosphate group, which is shaded green, and that phosphate group is carried on a serine residue in the active center of the enzyme. So that's a serine residue carrying that phosphate group. And that phosphate group is then transferred onto the glucose 1-phosphate to give us glucose 1,6-biphosphate. There was a first phosphate group on carbon 1 before you now have the second phosphate group that is coming from the enzyme phosphoglucomutase coming onto the 6th carbon. And so you have glucose 1,6-biphosphate. Now, in the second reaction, this phosphate group, which was on carbon number 1, is now transferred onto the enzyme phosphoglucomutase onto the serine residue in the active center of the enzyme. So you now have glucose 6-phosphate. And the enzyme is now carrying, this phosphate group, which goes onto the serine residue of the enzyme, is now, ready to undergo the subsequent reaction, the next reaction. That is phosphorylation of the next glucose 1-phosphate. So this reaction continues on and on and on in a cycle. Now, we see that in the synthesis of glycogen, we have the participation of nucleotides. And in this case, the specific nucleotide which is involved in the synthesis of glycogen is uracil phosphate. So you can say this is a uridine phosphate. You have the uracil, which is the nitrogenous base bonded on the 5-carbon sugar, the ribose, which then carries the sugar, the glucose 1-phosphate. So you see, what happens is, these nucleotides act as carriers of the sugars. So one after the other, you see the nucleotide, this uridine phosphate, carrying the sugar, the phosphate, the glucose 1-phosphate, one after the other, and they will be joining to form the glycosidic bonds between them. We'll look at that as we go forward. Now, what happens in the first place is that you see the sugar phosphate, that is the glucose 1-phosphate. The phosphate group on the sugar has this oxygen, which is carrying a negative charge, having lone pair of electrons, which has some nucleophilic attack on the alpha phosphate, the first, phosphate group on the nucleotide triphosphate. And that nucleotide triphosphate, the base there is uracil. So you have a uracil. So in place of this, you can say uridine triphosphate. Right. Now, this uridine triphosphate, so we just represented this to say, okay, this is the base. In place of this base is uracil. You have a ribose and you have 3 phosphate groups. But there is that nucleophilic attack from the sugar phosphate on the alpha phosphate group, which means there is going to be a break off of these 2 phosphate groups, a diphosphate or a pyrophosphate. We saw the term pyrophosphate earlier. So you see a pyrophosphate broken off, and you are now seeing the conjugation of the sugar phosphate to the uridine phosphate. And this is what you see. The full structure is what you see here. You see the uracil, the 5-carbon sugar, the ribose, and then you see the phosphate group, this phosphate group, which is here. That is the alpha phosphate group. That gives you a full nucleotide, which is carrying the glucose 1-phosphate. So this is the phosphate group on carbon number 1 of the glucose. So you see this whole nucleotide carrying the sugar phosphate, one after the other, so that they get joined together, forming glycosidic bond between them in that synthesis of glycogen. Now, we look at it further. We have the enzyme glycogenin, which is responsible for that polymerization of these glucose molecules. So this is what we were looking at earlier. Uracil, ribose, carrying, you have that uridine phosphate carrying the glucose 1-phosphate. So you now have that complex of uridine diphosphate glucose. So we see the enzyme glycogenin, which is responsible for the initiation of the polymerization of the glucose molecules. And at the active center of glycogenin is tyrosine. And you see that tyrosine is being written tyrosine 194, which I explained earlier, that if you see amino acids written this way, what it means is that if you take the sequence of amino acids in the peptide of that enzyme, the enzyme is a protein. So if you take the sequence of the amino acids in that polypeptide chain, tyrosine is the 194th amino acid in that sequence. That's what it means. And tyrosine is one of the aromatic amino acids. So you see that, benzene ring with a hydroxyl group. And the hydroxyl group, having lone pair of electrons on the oxygen of the hydroxyl group, has a nucleophilic attack on the first carbon number 1 of glucose, breaking off the phosphate group. And then you now see that glucose molecule is having some, call it, this is a COC bond, and this is some form of an ether bond between these carbons. So you now have this, or you can, so that is some bonding of the glucose molecule onto the active site, the tyrosine molecule in the enzyme glycogenin. So that becomes, the primary glucose molecule that binds to the enzyme glycogenin. Now, you see that this first molecule of glucose, the hydroxyl group on the 4th carbon the oxygen of that hydroxyl group also having a lone pair of electrons can have a nucleophilic attack on the first carbon of the next glucose molecule, which is carried again by that nucleotide, the uracil or uridine diphosphate. So you have glucose phosphate, the glucose 1-phosphate, and then you have the uracil, the uridine phosphate, coming together to give you the uridine diphosphate glucose complex. So like I mentioned earlier, all what it means is that you see the nucleotide carrying the glucose molecule and bringing it close to the next glucose molecule for the formation of the glycosidic bond between the two molecules of glucose. So it means this glucose molecule can now get, conjugated onto the first glucose molecule to give you that, glycosidic bond between these two molecules. And that, of course, we saw the formation of glycosidic bonds, and we refer to such bonds as 1,4 glycosidic bonds or 1,4 glycosidic linkages. The enzyme that is responsible for this, this is the initiation process, which is glycogenin. But after that, you will see that after the 8th glucose residue, you have glycogen synthase, taking over in the elongation process of glycogenesis. So now we see that elongation process where we see the enzyme, the glycogen synthase taking over. And this time, again, you see that you have that uridine diphosphate glucose complex, that is the nucleotide carrying the glucose 1-phosphate next to a disaccharide. So this is already the 2 molecules of glucose which have a glycosidic bond between them. And then there is a 3rd glucose molecule which is carried by the nucleotide, which then also gets bonded to the disaccharide to give you a trisaccharide. And that process continues on and on. That becomes an elongation process. And the enzyme which now takes over, as we said, from after you have the 8th molecule bonded in the initiation process where you are having glycogenin in that initiation process of glycogenesis, you have glycogen synthase taking over in that process. Now, you remember that when we were looking at the structure of carbohydrates in first year HND biochemistry, we talked about, polysaccharides, and we could have homopolysaccharides, heteropolysaccharides, and we also talked about branched and unbranched homopolysaccharides and heteropolysaccharides. Now, glycogen, as you are seeing it now, is a homopolysaccharide. Homopolysaccharide because it is made up of the same type of monosaccharide, which is glucose. So it's a homopolysaccharide. And it is branched. So it is a branched homopolysaccharide. And of course, if you are having a storage of energy, storage of glucose molecules, you need the branching to be able to store as much as possible more molecules of glucose. You increase surface area. If it is going to be just a straight chain of glucose molecules together, you will not be able to store much of that. But if you are having a branched molecule, you can store much more of glucose molecules in glycogen. So you have also the process of branching. And the branching which occurs in glycogen is the formation of the 1,6 glycosidic bond. That is the bond between the carbon 1 of one glucose molecule and the 6th carbon of another glucose molecule. That is what is referred to as a branch point, the alpha 1,6 glycosidic bond. And the enzyme that is involved in such process is being referred to as a glycosyl 4,6 transferase. 4,6 transferase because you have, on the very first, the main chain which is formed, you have the 1,4 bond being broken, which means you have the chain broken away from the 4th carbon of one of the from, let's say, that is the reducing side of, or that is the non-reducing end of the glucose of the polysaccharide onto the 6th. So it's like a transfer from 4th carbon to a 6th carbon. So if you break from here, you are breaking from 4th carbon. And if you transfer it here onto a 6 carbon, that is from 4 to 6. And so you see this is being referred to as amylo 1,4,2,1,6. So you have moved from 1,4, the alpha 1,4 bond, to form the alpha 1,6 bond. And so that enzyme is being referred to, the glycogen branching enzyme is being referred to as amylo 1,4,2,1,6 transglycosylase. And then you can refer to it as 4,6 transferase, glycosyl 4,6 transferase, as I was explaining earlier, because you are transferring from the 4th carbon to the 6th carbon. So sometimes you can also refer to that as a glycosyl 4,6 transferase, or you can say 1,4 to 1,6 transglycosylase. Now you see that, you have these chains. In each of them, you can have up to about, 11 to 12 residues. And, the transfer that occurs can occur in a fragment of, 6 or 7 glucose residues. So if you count up to, say, 6 or 7 glucose molecules, you can have a break, and then you have a branching. Let's look at that much more. So what I mean by that is that, you see, this is the very first chain which was formed on glycogen. So this is glycogen, the enzyme. The whole structure you see here, this is to show you the structure of glycogen. So that you have glycogen, and you have the very first chain, which is referred to as the primer. And on the primer, you can have 2 branches. And, such branches, they will occur maybe from the 6th or 7th glucose molecule. But each of these, branches can have between 12 to 14 glucose residues. And I think you have seen this in first year HND biochemistry. At least you have the structure of glycogen. And you can have this branching occurring, these 2 branches occurring on each branch, all the way up to having what we refer to as 12 tiers. So what it means by 12 tiers is this is a primer. And so the first branch will become, tier 1. And the branch which is on that first branch, those branches become tier 2. And this green coming on the second will be what? Tier 3. And then on that tier 3, you now have tier 4. So you can continue having those branches all the way until, up to 12 tiers. And in such a process, you have in each of these branches, you are having 12 to 14 residues of glucose residues. So in total, a full mature glycogen particle, can have up to about 55,000 glucose residues. So that's a lot of glucose molecules, a lot of energy being packed together, such that when the body needs energy at a time when it is in, a fasting mode, some starving mode, the body can fall on this storage of glycogen, breaking off 1 glucose molecule at a time to give you energy. So let's move on to the process of glycogenolysis, which is the breakdown of glycogen. So that will be like the opposite of the synthesis. In the synthesis, you are having a formation of glycosidic bonds. But in glycogenolysis, you are going to have the breakdown, you are going to have the breakage or the cleavage of the glycosidic bonds between these monosaccharides. So if this is a polysaccharide having, some glucose molecules with some glycosidic bonds between them, these glycosidic bonds are then going to be broken down one after the other. So you see the first glucose molecule is broken off, and that occurs under the action of the enzyme glycogen phosphorylase. And in that process, you have inorganic phosphates. An inorganic phosphate having an attack on the glycosidic bond, on the non-reducing end of the glucose molecule. That is on carbon 1. And so you now have glucose 1-phosphate breaking away from the chain of glucose molecules. So one molecule is broken off, glucose 1-phosphate. This is broken off. And when it is broken off, glucose 1-phosphate, it can undergo that first process that we saw, the conversion to glucose 6-phosphate. And that glucose 6-phosphate can go into the process of glycolysis and then generate energy. So you now see that happening with the breaking of the glycosidic bonds. But there is also branching. You see that there was branching in glycogen. So there is the need to break the 1,6 branch points as well. And that process is what is referred to as debranching. So you see from this is a glycogen molecule. And you see that the glucose molecules breaking off one after the other, giving you glucose 1-phosphate molecules. And you see there are some, you have from the branch point, the first molecule which forms the 1,6 glycogen, the 1,6 the 1,6 bond between these two glucose molecules. You see the first 3 glucose molecules which are bonded to it are shaded black. And that is because you have this enzyme, the glycogen phosphorylase, which is breaking off the glucose molecules, not able to break these glycosidic bonds between these first 3 glucose molecules, which are bonded to that primary glucose molecule on the branch point. So at that point, you see that a different enzyme, comes in to break off this molecule, a debranching enzyme, oligo 1,6 to 1,4 glucan transferase, which breaks off these 3 molecules and transfers it onto the main chain of that polysaccharide. And then you see that the glycogen phosphorylase takes over and starts to break off the glucose molecules one after the other. Then you have also the debranching. You have the glucosidase, which breaks off that branch point glucose molecule, which you see here. And then you have the glycogen phosphorylase taking over and breaking off these glucose molecules, breaking the glycosidic bonds one after the other. And once the glycosidic bond, is broken, giving you glucose 1-phosphate, the glucose 1-phosphate gets converted. The very first reaction gets converted into glucose 6-phosphate, and it goes into the process of glycolysis and then the TCA cycle, generating energy for the cell. That is glycogenolysis. Then the very last pathway is the pentose phosphate pathway. So pentose phosphate pathway, you see again from the beginning is glucose 6-phosphate, which means that glucose 6-phosphate, which you saw at the beginning of glycolysis, can go into different pathways. It either goes down the pathway of glycolysis, or it can go into the pathway of this pentose phosphate pathway, and it can also go into the pathway of glycogenesis. Can go into different pathways. Now we are looking at the pentose phosphate pathway, where we see glucose 6-phosphate, first undergoing some reduction, oxidation reaction, becoming a 6-phosphogluconate, and then there is some decarboxylation, and there is that formation of ribulose 5-phosphate, and then further to ribose 5-phosphate. And ribose 5-phosphate you have seen in the structure of nucleotides, which means this process of the pentose phosphate pathway goes into the synthesis of nucleotides. And of course, we also saw coenzymes such as the nicotinamide dinucleotide phosphates. We saw the structure of the flavin adenine dinucleotides, and even to some extent the coenzyme A, or coenzyme A also has some nucleotide present in there. And of course, these nucleotides will go into the synthesis of nucleic acids, either DNA or RNA. Now, in these processes, in this first process, you see clearly that there is that, can say synthesis of NADPH, because that is NADP plus, which is getting reduced, which means the glucose 6-phosphate gets oxidized. And you see that this then the hydride, which is picked from the glucose 6-phosphate, goes into what is referred to as reductive biosynthesis. This is some kind of a conjugation reaction where you have glutathione getting conjugated onto some either endogenous substances or even some xenobiotics. So it can go into drug metabolism. It can go into, for instance, even the metabolism of hemoglobin in the body, where you can have the conjugation of glutathione to, say, bilirubin, in order for it to become more water soluble, to be excreted out of the body in urine or even in fecal matter as a stercobilin. Now we also see in the second reaction here, again, further reductive biosynthesis this time around goes into the synthesis of some fatty acids, sterols, etc. But this is happening in what is referred to as the oxidative pathway or the oxidative phase. In the non-oxidative phase. So this is what happens in cells that do not, have this synthesis of nucleic acids or, nuclear material. In some cells like the red blood cells, for instance, red blood cells, just have hemoglobin in them to carry oxygen to the tissues. It doesn't need a lot of instructions. And so it does not synthesize nucleic material. So you also have, say, for instance, platelets. Platelets are just responsible for, say, blood clotting. It also does not require the genetic information, giving it much of information. So there's no synthesis of nucleotides. In such cells, you have this ribulose 5-phosphate getting recycled to glucose 6-phosphate. And in that process, remember the ribulose 5-phosphate is a 5-carbon sugar phosphate, and glucose 6-phosphate is a 6-carbon sugar phosphate. So you see that the the recycling to glucose 6-phosphate actually occurs in this is 5, so that occurs in 6 cycles. So 6 cycles, meaning 5 carbons here, 5 carbons. So 5 times 6 will give you 30. So some 30 carbons, and this is 6 carbons, meaning that 6 molecules of ribulose 5-phosphate will give you 5 molecules of glucose 6-phosphate, because 6 molecules here times 5 will give you 30 carbon atoms. Whilst if you have 6 cycles, meaning 6 cycles times the 5 carbons here will give you 30 carbons. So you have that cycling occurring, that synthesis of the glucose 6-phosphate from ribulose 5-phosphate occurring in 6 cycles. So that is the very last pathway of the metabolism of carbohydrates, which is the pentose phosphate pathway. Now I saw one question, somebody put a question that what is the purpose of branching in glycogenesis? So I mentioned, so that is Musa. I mentioned that the glycogen molecule, which is a storage form of energy, storage form of glucose, either in the liver or in the skeletal muscles, needed to have as much as possible more glucose molecules stored together. You need a lot of energy packed together. And so in order to have more molecules packed together, you have that branching in the molecule. If it was only one chain, you would just have a few molecules, together, and that is it. But you need to pack a lot of these together. And it is that branching which allows for the packing of these molecules. It's just like when you come to fatty acids too, you see you have, fatty acids and glycerol packing. So you have fatty acids packing on one glycerol. And then you have that, a lot of, carbon atoms being packed together. Same thing is happening here. You need this branching to be able to store as many glucose molecules as possible. Of course, it also increases some, surface area in a way. You realize that the breakdown of glycogen will require water. And so the branching of it will also allow for, some retention of water in that molecule, so that when there is the need for the breakdown of the glycogen into the single glucose molecules to generate energy in the process of glycolysis and the Krebs cycle, there will be enough water also being stored in that glycogen molecule for that breakdown. So these are the two things which you can refer to as the importance of branching in glycogen. That is, one, to be able to store as much of glucose, you need that branching. And it also increases surface area to be able to also have some water retention, which is required for the breakdown of the glycosidic bonds in the glycogen molecule. So that's the only question which I have seen in the chat. We have exhausted. I think we have how many minutes more? I think we should be having. We have 20 minutes more. So anyone with any questions, can put up his hand, and then I'll call your name. You unmute yourself, and then you speak. Any question or any contribution, anything you want to say. So the floor is open. We are listening. So of course, this lecture is being recorded. So after the lecture, I will send a video to the class rep to share with you. And as usual, I will also send the presentation slides to the class rep. And this time, I think I'll add a book chapter to it so that you can use all these materials for your revision. All right, so any question? Any question, any contribution? All right, the place is so silent. It's for 1 full minute. So I guess that no one wants to say anything. So I propose that we bring the class to a close, and then I will send the materials through your class rep to you. So till we meet, I hope that the next time, I think the exam is ending this week, so the next time we'll be able to meet face to face. So all the best and bye for now.
Bye.